Then alternate is not necessarily essential. It's called alternate sigma factor. And alternate sigma factor, I gave you some of the names. And those alternate sigma factors, they are responsible for this type of stress response. These are nothing to do with the housekeeping response. And so it increases or enhances functionality of those genes which can be affected by, say, heat, acid, all kinds of external factors. Okay? And so the sigma factor always increases the specificity. Okay? And also at the termination, the separation from the DNA, that also helped by this sigma factor. Okay? And so those are already described in those slides I gave you. So read up that information. And this is going forward. This is going to be an extremely important understanding. If you, like I said, in order to understand why microbes showing resistant to antibiotics, right, or resistant to certain phenomena, other factors, and this will help you to understand that better. Okay, if you can regulate this sigma factor function, maybe you will be able to control their physiology, their action, whether it's for disease or for any purposes. And so that's why this sigma factors, knowledge of sigma factor is extremely important. Everybody clear? Okay. Any other questions? Go ahead. Okay. So what is going to regulate, right? Well, that's a good point, though. When we said that there is no nutrient, right? Nutrient starvation, or pH, or salt, any of those external factors, or antimicrobials produced by other microbes, they can also regulate the QS synthesis as well. Okay, so that means that can affect the communication between cells or among the cells. So the production of the QS molecule may be affected depending on what conditions they are in. So if they're in a healthy environment, but there is lack of stress, they may not need to produce it, right? We all agree, when there is a stressful conditions, that situation is very adverse, they cannot survive well. So under that conditions, they would decide that I need to produce, a QS molecule so that I can communicate, let everybody know this is the situation. And so how do we handle or overcome these kind of adverse conditions so that everybody benefits in that community? Okay. Well, so yes, I mean, I guess if you don't like for them to form biofilm, right? And so they are forming biofilm, they are communicating each other also, this molecule. So if you disrupt the QS production, or there is an anti-QS molecule, which unfortunately I didn't tell you all of those. I'm waiting for the last day or day before. We have how to control biofilm formation. We got a lot of information. I'm going to talk about that. Yes, you will be able to control or regulate it. So the organisms which produce it, and if you limit their growth, or if you prevent them from being part of the community, they will be producing less EPS. A number of ways you can control that. Okay. Go ahead.
So does sigma factor only identify that cell is under stress condition or normal condition or any other?
That's one of the major indicators. So if you look at you can analyze the sigma factor in a gel, or you can see the proteins which are regulated by sigma factor. If they are increased, then you know that cells are under stress. Make sense? So they will produce number of different types of proteins or protein factors. I can say that acid resistant protein, for example, that protein in response to acid resistance, they will synthesize that one. So if you look at if the cells are under stress, they will make large quantities. If they are not under stress, then you will see their level is pretty low. No, I think that's an indication. This is like if you have fever, for example, if you have a fever and you cannot say which organism is causing it, right? Fever is a symptom of many type of infection. You see this? So that way, so if sigma factor is upregulated, you cannot say that this gene is going to be upregulated. Or if you find a gene product is upregulated, you cannot just say that this is the only thing happening, other things can be happening. Multiple factors can contribute to that function. Okay. Anything else? Okay, let's talk about, so let's finish that culture cell model. So we talked about the animal models and organ culture and the cell culture we are going to talk about. Any questions that we have? So these tools I've been mentioning, definitely these are powerful tools for studying pathogenesis, right? I think I need to go back and okay. Do you see this? I didn't talk about this. Have you seen this before? Called house shrew. Have you seen that? They're called house shrew. I think many of you may have seen it, right? So at home they would be around, right? They run very fast. So these are not mouse. These are pointy. They have a pointy face, correct? And they run very quickly. I've seen it when I was a kid. The last time I think I've seen it, the room I've been in. So I think this one just ran by very quickly, and they run very fast. I've never seen a mouse. They run. Okay, which one? Oh, okay. Go ahead. Okay. Anyway, so that house shrew, usually that's generally don't use for, or I would say not widely used as a model, okay? But I did put it there for there's some reason. One reason is this, is that you heard about the Staph aureus food poisoning, Staphylococcus aureus enterotoxin, they cause vomiting. Do you know that symptom, right? So anytime you have this toxin in food and within 30 minutes to 1 hour, you start vomiting, right? Have you experienced that type of symptom? Some of you, right? Don't deny. We all experience, especially in Indian food situations. A lot of times we encounter that. Back in US also we had that. So the whole idea being is that this vomiting is the main symptom of that toxin, and large number of people suffer from it, right? Because it's easy to be transmitted to food, because by handling, Staph aureus everywhere, right? And under right conditions, they will produce that toxin. So the one of the symptom of the toxin is vomiting, right? Or emesis, emetic toxin, we call it. None of those animals except pig, you know pig vomits? Have you seen that? Or if you don't know that, pig is one of those animals like humans, we vomit, right? But not all animals can vomit. And so within the rodent family, none of those rodent models they can vomit, or they can replicate the emetic toxin or emetic effect what we experience. So what they use, actually they use this house shrew. House shrew, this animal can vomit, okay? So that's why for Staph aureus enterotoxin study, they use this animal model to determine if the toxin you are using is a vomiting toxin or emetic toxin, got it? And we have isolated scientists isolated over 26 different serotypes of Staph aureus enterotoxin, okay? And one of the criteria, there may be more toxins produced by this organism, but one of the criteria to be a Staph aureus toxin, make sure they test that using this house shrew. Everybody clear? That is the reason I wanted to point out, because all those other animals are used for different purposes, but this one only used for vomiting testing, okay? So just, I thought it's a fun, because house shrew is very unusual, uncommon rodent member, and we use them for as an animal model. Okay, so we talked about different animals you can use. And we talked about if the animals are not available, you can sometimes use organ culture as a substitute for animals, and then that can help you to study. Now we are going to talk about the cell culture, and cell culture model is very common, okay? And because we don't need to use the whole animal, we are relying on the cells from specific organs or tissues, right? And that will be easier. So there are two types of cells, okay? One is called primary cell line, other is called secondary cell line. And by definition, probably you all know what is primary cell line. Primary cell lines are like if you grind up kidney or grind up liver, right? So immediately you are going to say, you can make a cell suspension of liver cells, okay? Then those cells are called primary cells, okay? And the secondary cells, which are actually a cancerous cells, okay? The problem with the primary cells, they have a short life, right? How long they are going to survive in a flash? Come on, you all know the answer. We talked about it yesterday. Close to 10 to 12 days or 2 weeks, right? That's their lifespan, primary cells, always, if it is not nerve cell. And so that's the general rule. So when you harvest these cells and make a suspension, and you can study these cells. For example, if you are trying to study a hepatitis virus or other pathogens like a Vibrio, and I want to use hepatic cell line. The reason I want to use hepatic cell line because these pathogens attack liver cells, okay? And so you can use this cell line to study their pathogenesis, whether it's adhesion, invasion, or cytotoxic effect, or apoptosis, or any kind of damage it may induce. You can study by using those primary cell line. Got it? But the problem is you have to use it within that 10 to 12 days or 15 days window. Okay? After that, they are going to die. The next time you need it, you have to use it again. So that's a problematic, right? And problematic also, you need to have a live animal to harvest those cell lines. Right? They want to join the class? No. So for that reason, primary cell lines are not going to be useful. So we need something more permanent, right? And that's when we started using the secondary cell line. And when you say secondary cell line, they are derived from cancer cells. Okay? What is the purpose? So that they are called immortal cells. Cancer cells are immortal, right? And so those cell lines, we call it cell lines. They are no longer cells. We call it line. Line. Whenever you see this word cell line, that relate to cancerous cells or secondary cell line. Is it clear? So the advantage, they are immortal and you can culture them in the laboratory. As long as you provide them food and nutrient, you can maintain them forever, like cancer. See this? And so, and then what happens, originally there are a lot of this cancer cell line already established. So for example, KECO2, I think we use that in our experiment, or people use that more commonly. And that cell line actually colon cancer cell. Colon, it comes from colon cells. So it's called Caucasian colon cancer or something. That's what the KECO comes. CA comes from Caucasian, means some white guy, person died, and then the next one is the CO, stands for colon. Okay? And so that cell line is widely used, but that's the picture of that KECO2 cell line, and this will grow, but it's never going to die unless you provide nutrient. Okay? So that's been commonly used. So you don't have to have an animal to harvest this. You can purchase it from the one of the major sources, ATCC, American Type Culture Collection. They always maintain it. Okay? And so this is generally what you get. So secondary cells, as you mentioned, they are transformed or hybridoma cells, and they have, if it is a transformed, that means they have a viral gene, so called SB40 or Epstein-Barr virus. These are known to cause cancer. Okay? And these genes are introduced into the cells to make a transformed cells. And hybridoma cells also secondary cells, and you can create hybrid cells, right? You have two types of cells, you can fuse them, engineer them to fuse it, especially for lymphocytes. They make that. Whether it's a B lymphocytes, which makes monoclonal antibody, or T lymphocytes also for studying a lot of different diseases. So those hybridoma cells are also called secondary cell line, and they are going to maintain a lot of their physiological attributes, including if they are immune cells, they are going to maintain their immunological properties. So that's the advantage. A lot of study you can do using these cell lines. And these are some of the examples of those different type of commonly used cell lines. HepG2 is from liver. If you need the larynx cells or lung cells, you can use Hep2. Henle 407, this is jejunal cell or intestinal cell line. HeLa cell, this comes from cervical cancer. The female cervix or cervical cancer cell line is the HeLa one. And J774 is called macrophage cell line. Vero cell, which is called monkey kidney, usually is from African green monkey, their kidney cells, okay, and these are called Vero cells. The Vero cells are highly sensitive to virotoxin. Virotoxin is actually toxin produced by E. coli. E. coli 157H7 or some of those, the toxin which damage kidney function, and we tested using the Vero cells. So the cell line help you to assay for many toxin or pathogenic interaction. The last one is called Chinese hamster ovary cell. So clearly it's ovary cell line. So all of those cell lines has epithelial characteristics. They look like epithelial cell, okay, or the macrophage cells. All those different types of cell line can be used. So now think of all those different diseases, different things we talked about. Essentially you can find cell line to study their pathogenesis, right? Yesterday we talked so much about pathogenesis of sessile versus planktonic cells, right? And what did we use? We used this cell line. Then of course we use the live animal, okay? And so this is how we design our experiment. So if you are designing experiment as a graduate student or researcher, a lot of times we study first with the cell line, okay? Once we know the data, if it works, then next level would be animal model, right? Then we use animal model and we verify, prove that this, whatever our hypothesis was, it works, right? Or you came up with a drug, or you came up with a vaccine, you tested it in animal model, and then you found the data. The next level would be human, right? We call it clinical trial. So these are the stages of research, what you see when you see the application in humans. So before it being anything being tried on humans, we go through all these steps. In vitro, cell culture model, then rodent model if you have, then we go through the farm animal model like swine model also used, then we go to human model. Everybody clear? And then after that we say definitely it's going to work or not. This is no matter where, what you are doing, always any kind of research application, everybody use these tools. So these are like a common tools, like your computer in your office, right? You cannot do any work right nowadays without computer. You cannot do biomedical research a lot of times without these tools. So these are powerful tools. Okay, so let's talk about how do we culture them, how do you grow them, what are the specific requirements. So this is KECO2, of course. This is a kidney cell or Vero cell I mentioned, and these are lymphocyte cells. Okay, these are B lymphocytes, but these are actually hybridomal lymphocytes. So if you look at B cells, this is what they look like. They are in suspension, and you can see this nucleus, and then the the cytoplasm, and then the membrane. And all these cells look nearly healthy. This may be in trouble, have some problem. This may be, but generally they look really bright. This is definitely dead cell. So by looking at these cells, you know which one is live or which one is dead, right? And that's the powerful tool you can use. I'll show you some more data. So how do you culture them? Remember, these are all human or animal cells. These are like us, right? And if you think of us, what we need for our growth, okay? So we need hormone, for example, right? We need amino acids, we need sugar, we need all of those components what is needed to survive or to maintain our health. cell lines also need that. And those specialized media are available. I don't know whether you have any cell culture facility on this campus. Anybody has that? That's excellent. So you already got good idea how it works. But for those who haven't have exposure to this, just to learn that there are specific requirement for this maintaining cell lines. So if you have to have a cell culture incubator. These incubators are specially designed to maintain certain gas level, say like carbon dioxide, which is essential. Even though we depend on oxygen, but we still need carbon dioxide for breathing purposes, right? So anywhere from 5 to 7% CO2 you need. You have to be humidified, temperature controlled, and that there is a water bath or water pan to maintain that humidity, and they will continuously circulate that. And these are usually pretty well controlled, maintained equipment. So these are the first requirement. And when you look at their media or the food, you can buy some commercial media, and they already have these essential amino acids, sugar, salt, buffers. And then the most important item is that called fetal cap serum or serum. And serum is the one which is essential. Without serum, you cannot grow these cells. And if you look at the serum, serum has a lot of micronutrients, right? Serum has hormone. Serum has a lot of growth factors. Serum has a lot of cytokines. So a lot of those factors needed for the growth of cells, and you can find it all in this cap serum. And they use cap serum because it's the young, right? And it doesn't have a lot of immunoglobulin, because when you become adult, our serum or our blood carry a lot of immunoglobulin. And those immunoglobulins means antibodies can attack a lot of organisms, right? Because they are against certain molecules. And so if you have too much of those immunoglobulins, they might attack those cells you are trying to grow. So that's one reason. And then also they have lower level of certain cytokines or growth factors. So for that reason, always we have to use cap serum or cap serum. Okay. And then it has also indicator called phenol red. And essentially when they grow in this cell line, the cell line grow in this media, they're going to utilize the sugar, right, carbon, or they're going to produce byproduct, right, which is acid. And if you have too much byproduct made, that would be toxic, right, because they are all in small flasks. And if you leave it too long, the cells are going to die, and you don't want to kill them. So that's why you have this phenol red indicator called acid indicator. So usually the media color is pink like this, and then if it grows up too much yellowish, then you know it's time to replace the media or do something, otherwise you're going to lose all the cells. So this is how whole thing is made. And fetal cap serum, usually I think the story behind in US we get this quite a bit. Usually the calf, the day it's born, usually the next day they will sacrifice this animal. They will collect the blood. And I don't know what they do in India, but in Western countries or other countries, they eat veal. You know what is veal? V-E-A-L. Have you ever heard about it? Probably none of you. Okay, I mean, it's not surprising. Usually it's very uncommon word. The veal is the meat of a calf. Okay, that's why it's called veal. It's very soft, tender. They market that. So veal industry is also a big industry in US. So what they do actually, they don't need also the male calf, right? Male calf are useless because they are using always dairy for milk, right? So those dairy cows giving birth to the male female, and so the male calf, they usually kill them. And they kill them, they collect the serum, because the serum industry is huge. There is a huge requirement. And at the same time, when they harvest the serum, they will use the meat. And so you can fulfill two markets in that. So the veal market is also pretty expensive market. Okay, and same time they use that serum for this purpose. So I don't know exactly how this industry works in India, because I don't think they do this type of practice here, and commonly people don't eat. Say that again. Just ABS? No, but it's still, that's from the adult cow. Adult, when they slaughter it, they collect they use that for ABS. You can use that serum But for these purposes, you need to have a cap serum. But you can use, like I said, adult serum, adult cow serum after slaughter, but those serum needs to be processed properly, and plus it doesn't have all those factors. So probably they can also import from other countries like US, usually very expensive. That's the most expensive item. So we buy a 500 ml bottle, because sometimes $1,000. Okay, so now you know how much it's going to cost. It's very expensive. The price is going up. So just to tell you the side story, because where this cap serum come from, why you have to kill the cap, is there any use for it? Anyway, so my goal is to always make you know a lot more, right? Sometimes you use cap serum, but you have no clue where they come from, right? Anyway, so these are some of those media. For if you pick up any of those cell line, the Hubei called, this is endothelial cell line. And there are some specifications, again you have these slides, right? Specifications of media. They formulate in certain way that this cell line, if you're trying to grow it, you should use this media, okay? So you already have this knowledge or this slide, so you could use this as a reference for future. And I use this also same way. So advantage of culture cells is pretty simple, whatever I just mentioned, and you can simple and control model to study host pathogen interaction or toxin activity. A lot of this cosmetic industry also they use this before they go to animal studies, right? How do you know it is a non-toxic? That's the first question. So you can use cell culture model first. And they grow rapidly, so it's easy to use them, convenient. You can try with toxin, pathogens, whatever it is, right? You don't have to worry about. It's not that animal. And then primary cells can be, like I said, you can use primary cells, no problem, but you have to harvest each time you need it, right? You cannot maintain them. And then because it's a primary cells, each time is going to be a different result. So it may not be reproducible, okay? But primary cells, I like it, there is a reason for it. Primary cells actually true representation of that organ or tissue you are going to use, because it has a mixed type of cells. Secondary cell line, only pure cell, that cell line. And the primary cells, you will have a say epithelial cell, then you will have a fibroblast, you may have some immune cells, macrophages, all those other cell type in that lung, liver or kidney you harvest from. So you can have all of these. So that's the advantage, but there is a disadvantage, right? So for that reason, you have to understand what you need, how you can use those for your research tool. Culture cells are derived from tumor cells. So that's the problem, right? Disadvantage or limitations. So their properties has been changed. They are not natural or normal. Okay, so that is one of the issues. So they may not be genetically identical. Continuous growth will produce mutations. That's another issue. They will be changing. So you cannot subculture all the time without thinking that they are not going to be changing. So they are going to change sometimes. So that might affect your result. So that's why you have to maintain certain passage number. So you grow them for a while, and then you stop that, then you go back to your original, and then you start all over again. And for maintenance, you have to also have a liquid nitrogen storage tank where you can keep those cell line for extended period of time. Okay, immortalized cells are different from the normal cells. Loss of shape, distribution, surface antigens. Culture cells are not polarized. This is another thing. You understand what is polarized means? When you say polarized means you think about North Pole, South Pole, right? So the polar means in the gut, epithelial cells, some cells are facing, certain properties, cellular properties are always upwards, right? And then certain things at the bottom. But when you grow them in the flask, you don't know which is top and which is bottom, right? Which is our outer layer, which is the bottom layer, right? So you don't have that. So that's the problem, you cannot have that. And so you have to make it polarized, and that means you have to use proper cytokine or growth factor, proper hormone. You can use it to make that happen, okay? And so you have to know that. So I will show you some examples. Yesterday, I think I showed you this KECO2 cells with the microfiber, like when Listeria trying to get in. Those are polarized cells. We made them polarized. Okay, you can do that. So that's the part of the technique. What else? The other thing you can do, because it's a single cell, you don't have all those other cell types, That's the primary cells you will have. Anyway, you can, with the secondary cells, you can add other components. You can add mucus, you can add other, antimicrobial molecules, a lot of things you can add to create or simulate like what it is in the gut or in the lungs or in the kidney. You can play with this. And nowadays, actually, a lot of people are using stem cell, right? So, everybody knows what is stem cell is, right? Stem cell is the cell where all the cells are originate, right? So, our bone marrow, for example, that's where the source of stem cells, okay? And so, if you run out of certain cells, the body actually replaces those cells using the stem cells. They can differentiate. So, yesterday, one of you had a question about the ear, right? Organ culture, right? We talked about, so usually a lot of times we start with the stem cell. So we put the stem cell in that culture media, then we have to use proper cytokine or growth factor, then it can differentiate into whatever the cell type you want. Because stem cell is the original cell, right? And it can differentiate into the type of cells you need. And you do that by adding proper cytokines or growth factors. Got it? So if you ask the stem cell to become a called epithelial cell or fibroblast cells, you can make them do that. You ask the stem cells to be immune cells, you can make them too. So you have to give them. So that's what we start with many times. The stem cell research is also extremely important areas of research. Anyway, so how do you assay it? So the measurement of virulence. So of course, when you are using animal models, certain way you can assay it, right? Assay them. So these are different way you can introduce your pathogens of interest. And then one of the things, if you are looking for death or life, it's called lethal dose 50. Are you following this part? So like if you want to know the pathogen or the toxin you are looking at or studying, how toxic is it? So usually you call it lethal dose 50 or lethal dose 100 or lethal dose 75. So that means 50% of animal going to die with that dose, right? So you do a different dosing. And so that's kind of like it's like a radioactive decay time, right? We call it half-life, right? Because you don't worry about how many years does it take to completely, decay the isotope or radioactive molecules, completely disappear, right? We always calculate by half-life, right? Say if it may be 14 years for, carbon C14, right? Carbon isotope or radioactive molecule. And so that's how also for lethal dose, we try to calculate or report based on the 50% or 75% animals. So that's one way you do this. And I think I have a plot for that. So this is how when you do the assay, whether you use any kind of animals, whether it's a C. elegans, the parasite, or you use the larvae or rodent model, and you can always do this type of plotting, okay? And so these are special called Kaplan-Meier survival curves. This is always used. So if you look at the different dosing, higher doses, the mortality would be higher, right? This is higher mortality, right? If you have a 10 to the power 10, 5 to the 10, and you can see the mortality is much higher, as the dose decreases, you can see the mortality decreases. And so this is called survival probability or percentage, got it? So this is the statistical program, we always use that. This is for some other ones. So these are some of the examples how we report the toxicity of or pathogenicity of a given organism or molecule or chemical. Everybody clear? You have to do it at some point, remember. Make sure you understand this, even if you haven't done it now. So this is cell model, I mentioned that. If you have a healthy cells, this is vero cell, right? These are like a elongated fibroblast like cell, and if it is exposed to E. coli toxin, which is the Siga toxin, or sometimes we call it virotoxin, look at it after like somewhere 48 to 72 hours, this will look like this. So you can look under microscope, right? And this exposed to this toxin and all these cells, do you think they are alive? Looks like they are all dead, right? So this is called CPE, cytopathic effect. Cytopathic effect. This is a terminology we use that. So this normal healthy cells converted into more round cells. They flocculate. They come to the surface. They detaches from the plastic. So these cells, epithelial cells, they are always attached to the plastic. That's how they grow. And then when they're dead, they can come off. They become granulated. They become easy to observe. So sometimes you don't need anything, just look under microscope. You'll know it. Sometimes my eyes also, sometimes if you are well trained, you don't need even a microscope. I can tell you just looking at this, I can tell all this certain percentage of cells dead. You know that it's toxic. Okay. So this is how we do it. Otherwise, you can stain them, stain the cells. It's called live death staining. One is the common one, it's called trypan blue. And so if you look at these clear cells, if you add the dye, this dye is very easy procedure. And if the cells are alive, they don't take the dye. If it is dead, the dye gets in. So this is live cell, this is dead cell. And if you have seen hemocytometer, the one we use it for blood cell counting, and you load it there, then you can count it under microscope. Then you get a live dead estimate. And this is an essential tool for immunological assays. They always do that. Or anytime you are doing cell culture, you have to know how to use hemocytometer to count the cells. These are not bacterial cultures, so you have to grow them on the plate, count it, not like that. This is like you have to put them in this counting chamber, hemocytometer, and you count this. Okay, you are either counting live cells, that's the best way to do, because a lot of cells would be dead, or you can count just the dead cells. Either way, you can calculate it. And then at the end, you can calculate the percent dead, right? It's pretty easy. Total number of cells you used, right, divided by the number of cells already live or dead times 100, right? So that's very, very easy. And you have to know the calculation, how many cells. If you find 10 cells, and it's like a 10 times 10 to the power 4, that's the calculation. It's very simple. And you don't need a calculator or anything. Just you give me the number, I will tell you this is the percent cytotoxicity. You can easily calculate it. So these are some of those other graphicals or pictures. So you can see this is control. If you add vero cells, if you stain it with the trypan blue, you can see this lot of dead cells. This is another control, maybe non-pathogenic E. coli. It's not killing it. These are some of the lymphocyte cells also respond to the toxins. This is already I mentioned that, right? These are all dead cells. These are still alive. These are lymphocyte cells. So you can also use electron microscopy to assess the cell damage. It's very simple and look nice. So this is B cell. B cell harvested from animals. I think this one, or I say human B cell. And when you look at the B cell from humans, they got a lot of the surface molecules. And if they are sensitive to toxins, a lot of those makes a big hole, and it loses all the small molecules. This is also more uninfected versus these are infected with Listeria monocytogenes. You can clearly see the damage. Other way, what is the other best way you can do? Those are like a microscopic or observational, but in this case also this is the most easy, and I like this. So you are measuring enzyme release, right? If the cells are damaged, the membranes are damaged, they will release their internal enzymes, right? And if you have a proper substrate, you can detect that enzyme. And the degree or level of enzyme activity corresponds or correlates to the amount of toxin you have or pathogens you have. Okay? So this is, you can use it called alkaline phosphatase. It's one of the enzymes many cells has. And then if you add the substrate, they will change the color. So if you, these are some of the substrates. So paranitrophenyl phosphate. Nitrophenyl phosphate is the substrate for alkaline phosphatase, right? And then when the phosphate is clipped off, the phosphate, the benzene ring, that's yellow color. Okay, that gives you the color. This is how you assay it. And these are really powerful tools. You can generate so much data for doing this assay. Sometimes you can also add proper substrate, so methyl amblyferol phosphate, and it can give you fluorescence, which have a higher sensitivity. Okay. The most commonly used lactate dehydrogenase assay, or called LDH assay. So the cells carry lactate dehydrogenase, right? If the membrane damage, the enzyme is going to get out, and you can add the substrate, and this pathway actually it converts tetrazoleum, which is yellow, to formazone, red, and this the pathway it goes lactate dehydrogenase convert lactate to pyruvate, and the NAD converted to NADH to NAD, and during that time this hydrogen ion obviously contribute to these changes in the substrate color, okay, and that color you can measure it, and that's how you know that LDH is released, like the alkaline phosphatase. People also have used the beta galactosidase. A lot of these enzymes assay can be used to assay for the actual action. There is also other assays called MTT assay. These are more metabolic activity. Other is called WST assay. These are also again determining the metabolic activity of the cells. Also you can do if the cells are going through apoptosis. You heard about apoptosis? All of you should know, right? It's a programmed cell death, and opposite is the necrosis, right? So one is called homicide, other is called suicide. Necrosis is a suicide, right? Killing themselves. And the other way around. Homicide is the necrosis, and the suicide is the apoptosis, programmed cell death. Do you know why cells do that? Do you know why they undergo apoptosis? You have some idea, right? Yes, some of you have some ideas. So we notice that many diseases, when there is pathogens or toxins coming into the body, so the cells try to kill themselves, right? Why? Because they don't want that gene to be propagated, right? So the apoptotic cells do not induce inflammation. What it means is that the macrophage will clear up those dead cells. So the genetic material is no longer has the ability to transfer to other cell, okay? And that's a different strategy. Whereas the necrosis, if the cells die, the gene or DNA remain intact. There is a possibility that other cell pick up the genetic material and then they will perpetuate that problem, okay? So a lot of viral infection or bacterial infections, we have noticed that this is the defense strategy. I don't want the cells when they are being infected by Listeria or Salmonella or some viruses, and they will integrate their genes, right? And we don't want that gene to be perpetuated, propagate in the body. So that's why if I kill myself, okay, and this body, the macrophage is going to come and just swallow it and destroy it, and there is no trace of those genetic material. Necrosis, on the other hand, if it happens, it induces a lot of inflammation. A lot of immune cells come in. Everyone coming in, they will try to clean it up, and in the process, that genetic material can be transferred and can be maintained. Okay. So cancer cells, do they go through apoptosis? No, right? So if you can find a way to induce apoptosis in cancer, then you got a cure, right? Unfortunately, those are you cannot kill them, right? They metastasize eventually, and so you cannot kill them, and that's the problem. They don't go through because they have the proper gene to maintain that status, right? So apoptosis, you can study this because this is a tool, right? Now you have to know when and how I'm seeing the cells undergoing apoptosis or necrosis, right? All of those changes. So for that reason, you need to know how do you assay them. Okay, so there is a different way you can do this, stain them. These are different types of staining you can use it. And annexin is one of the really powerful tools, annexin 5 binding assay. If I have more time, I would have explained you a lot of this stuff. But if you look at the cells going through the apoptosis, cell shrinkage, the meiosis stage, I will show you the pictures actually. I think I may have pictures. These are some of this data from our research. And you can see this, the cells, intact cell. If it is going through apoptosis, you will see the nuclear material eventually degrades, becoming a small fragment, and these fragments getting called bleb, and then eventually macrophages clear them up. These are the standard procedure or step. Okay, that's how this cells taken care of. And if you stain them by fluorescent or acridine orange, you can see these changes taking place. Okay, all of this, and then eventually it's making this bleb. So it's fascinating, and the reason again, what research you will be doing in future or whatever areas you may be, and so you may have to study, understand whether they are going through necrosis or apoptosis. Especially in cancer research, so well, I want to come up with a drug to treat the cancer, then can I induce apoptosis? What signatures you have to look for, right? The DNA degradation is one of the major output. Always DNA get fragmented. So that much I wanted to tell you. But if I am giving a lecture to my students, I will spend a lot more time to explain every steps of it. Unfortunately, you cannot dedicate that much, but I want to give you a flavor of what are needed or what we understand from this cell-based assay. Okay. We talked about adhesion, right? We talked about attachment quite a bit. How do you do that? That's very simple actually. You grow these cells, add your bacteria, and after some time, you just wash it off, like a 30 minutes to 1 hour. Then you assay how many of them are bound, whether you want to stain it or you want to plate it, right? Then you look for count, colony count. And this is how we know adhesion. If they are gone inside, how do you know they are inside the cell? So what you have to do, in order to monitor the cells inside, you have to kill the cells which are outside first, right? So you have to use antibiotic to kill outside cells, and then after you wash it, then you find out how many inside. You have to lyse the cell and get the bacteria out. And so those processes are very straightforward, and you can do this. These are pretty simple. You can again follow this, and that way you will know. And this is the cell, actually neuron, nerve cell I mentioned, keep on mentioning. These are nerve cells bound by Listeria. Anyway, and the other one you can see is that if the cells are passing through to cell layer, right, paracellular. And I showed you this picture before. This is also a very powerful tool for those who are studying gut health or gut permeability. This is what we do. You have a transwell insert. There is a filter membrane underneath, and you grow your cells. So for example, you want to grow KECO2 cells, you want to grow HT29, or any of those intestinal cell line, or even other cell types, and you grow them in a monolayer, and when they are growing in the monolayer, and you can measure their electrical impedance, we call it TER, translocated epithelial electrical resistance. What it means is that, how do you know they are forming the monolayer? It's very difficult to see them under microscope. That's why we have a probe, we measure that electricity. You all had physics, right? All of you, undergraduate, I mean high school level physics at least, not the college level. So how the electricity move? Negative to positive, right? And if there is a water in between, you have two electrodes, right? It will flow one type of resistance, right? Always resistance, right? And that's called impedance. Impedance means impede the progression. That's what we call impedance. Okay? Conductance means, I'm not an engineer, so I'm trying to teach you a little bit. So the electrical conductivity we are trying to measure. So the water will have one type of conductivity, right? If you add salt, you will have a higher conductivity, right? So the ions which are transmitting the electricity. And so here, if you put a probe here in this one and one outside, and then the electricity movement would tell you the integrity of the membrane. If these epithelial cells made intact monolayer, there will be high or low resistance. Low resistance, right? So that means electricity has opposite. This is where I get, it's not confusion. So if you have an intact cell monolayer, you have a high impedance, means high resistance. So we measure that, say, get close to 500 ohm, right? And that's the resistance. But if the cell monolayer is damaged, the resistance would decrease, right? Because the cells are not intact, and therefore electric current movement would be faster or quicker. Because this liquid is actually ionic molecule, right? A lot of charge there. And so based on that, we determine the integrity of this membrane. layer. Okay, and so that's the thing we do called tear measurement. So again, if you read it up, if you in future you need to use this type of tool, you'll find it interesting. And other thing you can do also, if you add bacteria, if they go through and they will be large number here. If somehow blocked, then you will not see them downstream. Other thing also you can use also dye, put some kind of fluorescent dye on the top and see after 1 hour or 24 hours later how much of the dye at the bottom layer. And that tells you the integrity of this membrane. The reason I'm spending a little time on this, because a lot of those diseases, even this heat, have you ever suffered from like a vomiting after coming from heat or diarrheal episode? You had any of you? It's a common practice. I think 2019, I came to India and I went to Varanasi, right? And that was happened to be summer. And I went to one of those parks to visit that area. And that day temperature was close to like what we were experiencing. That day I think it's close to 42 or 43, if not 45. It was a very hot day. And so we end up, I think we are kind of naive, not really paying attention to the heat. It was kind of like a dry heat. And so we spent like an hour or 2 hours going around, but we had umbrella and stuff. And then immediately in the afternoon, we started showing symptoms of diarrhea, okay? And especially me, and then I think one of my children also showed the symptom, but no temperature, nothing else. And we know that it's not coming from food, because none of the food we didn't eat outside, everything is well prepared. And the reason I'm telling you that this is the first time I experienced it. I never had that type of episode. And then realized we suffered for almost 2 days, continuous, and we're trying to replenish with electrolyte. Always have a lot of electrolyte, right? And then you study more about other people, those who are long distance runner. This also sometimes, you notice that when they're running marathon, sometimes they throw up, right? And also some people suffer from diarrhea. And these are non-infectious diarrhea, okay? So what happens, some of those stressful conditions, it makes our gut called leaky gut, okay? This epithelial lining in the gut, it behaves like it's infected by, say, cholera organism or Shigella or Salmonella. Shigella is different. So you lose a lot of fluid. So what it does actually, it opens up your gut barrier. So it makes it leaky gut. And this heat stress makes it much leakier. Heat, some of the stressful conditions, sometimes you know that people when they get really stressed or nervous or anxiety, they vomit, right? Notice that? It happens, right? A lot of those natural response. What happens, you can define it by saying that your gut is leaky, so all those fluid coming out, okay? And you can study that, you know, on this type of model to show that the leaky gut can happen. And I'll show you a little bit more data. But you can, in the laboratory setup, you can create that. So this cell, if you're looking at these two cells, this kind of separates out. I think I showed you some pictures, I will show you a little bit more. And leaky gut is common, is the main reason for a lot of diseases we suffer from. Name any disease, see if you can find a link with the leaky gut. Parkinson's disease, Alzheimer's disease, those are the serious diseases, right? Inflammatory bowel disease, all of those, heart disease. A lot of diseases are now they're finding that it's attributed to leaky gut. Why? Because of the microbial community we have, right? What microbes has? LPS, peptidoglycan, all those nasty surface molecules. So all of those microbial, billions of bacteria or microbes, their molecules being absorbed to our system. Immune system responds badly. And that's what we are finding out, the leaky gut is responsible for many diseases we suffer from. Even to maintain your own health, think about it. So I wanted to give you the science behind it. And so the leaky gut mechanism we are finding out is responsible for so many diseases. I have a huge list of those. I don't know if I have that slide or not, but I will show you those list of diseases come from leaky gut. And we usually study using this. Anyway, so these are the cell-based assays you can do it. And also you heard about the whole body scanning, right? Bioluminescence. This is the whole body bioluminescence imaging. And if you have a bioluminescent bacteria, you feed the animal, as the animal move from organ to organ or different places, and you can take a real-time picture. These animals are not actually dead, these are live animals. And you just anesthetize them for a certain period and take an image, and then you can bring them back. So that's why we call it live animal imaging. Like the human body, we do MRI and all of those live scanning, and these are actually bioluminescence. We put the marker to measure that. These are more studies done. So let me go back. One thing I wanted to highlight. How do you know you have a leaky gut? Usually, if you go to a gastroenterologist, if you have a intestinal problem, they will give you some kind of dye. And you will drink that dye, and after 1 hour or two, you can collect urine and look at the dye level in the urine, okay, or in the blood. And if you have a high level of that dye in the blood or the urine, that means you got a major problem in your gut. So that means you have a leaky gut. And that's where how to treat your gut so that you can close them. Not everything needs to be getting into our blood circulation. And so we do that, follow the dye marker, okay, just to give you the idea how we test for leaky gut. Anyway, I know it's already 1 hour. Any questions on this? Go ahead, Rohan. Use the microphone.
Sir, you mentioned the relation between Parkinson disease and leaky gut. Can you explain and give more insight to this? Because Parkinson is necessarily a neurological problem. It's a critical illness being discovered.
So again, I have to go back to the microbial components. Okay, they are taken up by the gastrointestinal tract. Eventually, get to the molecules. All those molecules get to the body, right? The blood circulation. Then you don't know exactly how the immune cells responding to each of those molecules. And so, if it is a neurological disorder, some of those molecules getting to the brain, and the brain also getting damaged. And same way with the Alzheimer's disease, is forming this plaque. A lot of those plaques actually derived from these microbial components are playing a major role. Okay, so varieties of chronic diseases, including heart attack or heart disease, also the stroke. People suffer from stroke, right? And forming this small clot in the blood vessels. And these are all we're finding out is the microbial origin. Because of the leaky gut plays a huge role. Besides the cholesterol or other molecules creating a plaque. So it's a huge. That's the current trend of research. That's the modern approach to finding out the causation or causative agents for, including arthritis. I think we mentioned arthritis other day. A lot of those molecules also antigen antibody interaction, and they're complex forming, and they're traveling throughout the body, and how immune system responding. This is a complex process, though. I'm trying to simplify it. Okay, but these are all linkage they're establishing. So I think it's a kind of an alarm, but I think same time it's better to know that because there are some recent review articles, they cover all of those things. If you want, I can bring those papers or send you the links so that you will study this, how a lot of those leaky gut, especially. The word leaky gut, as soon as you hear it, so you should be alarmed, am I keeping my gut healthy? So that's why I focus on gut health. How do you keep it healthy? How do you prevent the leakiness? Right? So I'll try to tell you some of the solutions. So that's why we take right nutrient, proper diet, right? And a lot of these toxic molecules, they cause this barrier to open up. I've been studying this leaky gut for a while. So the proteins, I showed you some pictures, right? How these proteins holding this cell structure together, and it's fascinating. The microbes manipulate it, or different conditions can manipulate this signaling cascade and open up the junction. And some organisms just wipe out that totally. So from infectious disease point, it's very important. Also from the chronic diseases people suffer from, or the type of diet you eat, that can also influence. A lot of factors can influence the gut integrity. Okay, and so it's not just one factor causing it, but the leaky gut is really an important concern. Always you should be thinking of how can I keep my gut healthy? Think about that. Healthy gut. Okay. I know it's time for break, right? It's 10 to 11. I think it'll 5 past, so we'll